I'm sure you know all about carbohydrate and lipid metabolism. But how about ketones and proteins? Welcome back to day 4 of MCAT 528 and 28. When the body is starved of glucose and the glycogen reserves are depleted, it can rely on fatty acids and proteins, our other two macromolecules, to compensate. However, both of these have their own issues. Proteins play a vital role in all of our metabolic processes, which is why we don't want to lose them and we don't want to lose the muscle on our bodies. Now, the issue with lipid metabolism is twofold. One, that the brain can't actually use it for fuel because lipids cannot cross the blood-brain barrier. And two, beta oxidation can only run for so long before acetyl-CoA and NADH accumulates, stopping the process via Le Chatelier's principle. Instead, the liver is able to take these acetyl-CoA, turn them into HMG-CoA, and then acetoacetate as well as beta-hydroxybutyrate, which are two very common ketone bodies that are able to be used by our brain and the rest of our cells. This is actually the idea behind the keto diet, whereas fat stores are thought to be deleted not because they directly contribute to our energy sources, but because the liver is able to turn the byproducts of beta oxidation into ketones, which are then able to fuel our body. Now, you might also want to know that the rate-limiting enzyme of ketogenesis is HMG-CoA synthase, and of ketolysis, that would be the enzyme known as SCOT. Now, diving a little deeper into the amino acid metabolism that I mentioned earlier, which actually accounts to about 10 to 15% of our total energy production, we can see that the alpha keto acid, formed from the deamination of amino acids, can enter either the carbohydrate or fatty acid pathways, depending on whether they are ketogenic or glycogenic amino acids. Now, the glycogenic amino acids can turn into pyruvate, oxaloacetate, or one of the intermediates of the citric acid cycle, whereas the ketogenic amino acids can turn into acetoacetyl-CoA or acetyl-CoA. This deamination step is actually known as a transamination, where alpha-ketoglutarate accepts the amine group from the amino acid and turns into glutamate, which can then enter the urea cycle and allow for the excretion of ammonia.